Welcome to Dementia Matters, a podcast created by the Wisconsin Alzheimer's Disease Research Center. It's our goal to humanize Alzheimer's research so that our community, our patients, our participants, and anyone else interested can get a better understanding of the work that's happening to fight back against this disease. My name is Nathaniel Chin, and I'm a geriatric memory clinic physician at the University of Wisconsin. I'm also the family member and son of someone with dementia. I'll be serving as your host for this podcast and asking the questions I believe are in the minds of many in our community. Thanks for joining us. Welcome to Dementia Matters. Our guest today is Dr. Howard Federoff, a groundbreaking researcher of brain disorders and a nationally recognized healthcare and science leader. Several years ago, Dr. Federoff discovered a blood test that predicts the development of Alzheimer's disease with 90% accuracy. Currently, Dr. Federoff is a professor of neurology at the University of California at Irvine. Dr. Federoff, welcome to Wisconsin, and thank you for joining us on Dementia Matters.
It's a pleasure to be here with you.
A paper that you published in 2015 that showed that a certain blood test looking at specific brain proteins could predict the development of Alzheimer's up to 10 years before onset and with this high accuracy. Now, for our listeners, can you explain what this study was and the results?
Yeah, sure. So in addition to the work that we were doing that relates to plasma metabolites, we became interested in understanding another part of what circulates in our body, and that is the so-called microvesicle, or people tend to call them exosomes. And what they basically are little packets that contain cargo, and some of the cargo could come from places including the central nervous system. We entered into a collaboration with another translational group to see whether we could ask and answer the question, is there an exosomal fraction, these little packets that contain molecules that are associated with Alzheimer's disease, like A-beta or several of the different tau isoforms, that were differentially abundant in patients that were part of our longitudinal cohort, including those that had entered when they were cognitively normal. And then, unfortunately, a small fraction of them would progress through a phenoconverting step to develop typically amnestic MCI, mild cognitive impairment, before going on to Alzheimer's. So the study was really kind of simple in design, was to use what I would call, in quotations, neurally derived exosomes, in that we believe that the method of their isolation, by virtue of what's on the surface of these little packets, would suggest that they're likely to come from the central nervous system. And then once purified, we were then able to analyze the content, including A-beta 40, 42, total tau, and several of the tau isoforms. And this was a collaboration with a number of other investigators. And the basic bottom line was that the exosomes contain these apparent CNS-derived molecules of interest that we often talk about as present in the brain and the CSF, and sometimes even free in the blood. And they were differentially abundant in individuals who were at risk for the development of Alzheimer's disease. So if there is a conclusion here, it's that there's a new frontier in signaling.
A new area indeed. I mean, all of these things sound very new, but also very complicated.
They are.
And so it's a process that's going to take a lot of research.
It will. It's not simple, for sure.
I use the word blood, and then you correctly use the word plasma. For our listeners, can you explain plasma and what that means?
Yeah, so plasma is often considered to be a fraction of the blood. From one blood draw to the other, you can get far more consistent measurements of a variety of macromolecules, including metabolites. Plasma turned out to be the right type of starting material in order to be able to reduce variability within a clinical center and across clinical centers.
And so then for people who are a part of research, one of the things I think is fascinating about what you're looking at is blood draws. You mentioned this in a clinical setting. So this is not someone who's getting a scan of their brain or going in for a lumbar puncture.
Right.
This is something that can be done relatively easily and quickly.
Correct.
So do you see in the future an ability for us to draw someone's blood and tell them in a clinic setting their percentage of risk of developing any form of cognitive issue?
I do, although I would stratify the question to be a little bit more precise. But generally, yes. I think it's likely that if you look at the life continuum, that the closer you are to the age, and we're talking about the more common late onset Alzheimer's, it's more likely that the specificity of the measurements will produce greater accuracy that could be conveyed to the patient in a clinical setting.
But it's very exciting, and it does sound like this is coming down the road.
I think so. And now our blood test, our metabolomic blood test, is about almost 97% accurate.
The research that you do with certain blood tests, you also are looking at the factors in the development of Alzheimer's disease. And I'm referring to a recent publication in the journal Neurobiology of Aging. And I think this came out last year.
Yeah.
And you looked at the blood of people, and I like this, with superior memory, normal memory, and those with cognitive impairment, both mild cognitive impairment
Yes.
and Alzheimer's. And you found noteworthy differences in, I'll call them components, 12 components or
Right.
factors.
Right.
And you found that these factors dealt with regulatory pathways in the body.
Indeed.
And so I'm hoping you could share with us what you found and how this relates to a concept called cognitive reserve, or our brain's ability to withstand insults or damage without actually showing clinical change.
Yeah, no, that's a great question. And so I'm going to sort of divide it into components. The first component really is, what can we learn from individuals who have very successful aging and who not only have demonstrable cognitive performance, but in some cases, because there is a sort of distribution in terms of how normal cognitive performance, adjusted for many factors, and that which may be superior need to be defined. We were not the first to define these very successful agers, but we found them in our cohort. And the team became very interested in understanding in the blood of those successful agers. We asked, is there a blood set of measures that would be distinguishing that group from normal, and then separately from those who have cognitive impairment, either MCI or AD. And we found these 12 metabolites, or these components, 6 of which are phospholipids and 6 are amino acids. And the interesting thing, as we sort of start putting this together as a story, is you start thinking about, if there is something in the blood that's telling you about cognitive performance relating to above average cognitive performance, then the blood is mirroring something now that is unrelated to disease. It's telling you something about cognitive performance, and therefore, to your question about reserve, I think what it's telling us is that if we can understand the genetic architecture that produces in the periphery, in the blood, as the plasma we talked about earlier, something that allows those individuals to sustain above average cognitive performance, that may be very helpful to be able to map back onto what kinds of therapeutic options do we have for patients who either have cognitive impairment or may be at risk for cognitive impairment. The interesting part of that study, which when I presented this to the group as an idea initially, many thought I was a little loony, but I said, well, bear with me for a moment, but if these 12 components can help us classify normal from the supernormals, could they be thought of also classifying those with cognitive impairment from normal? And we might even anticipate that the change in that particular component in supernormal, where it may be higher compared to normal, now might even be lower still in those with cognitive impairment. So they indulged me. We did the analysis, and in fact, it produced a very highly sensitive classification where we combined MCI and AD patients compared to subjects with normal cognitive performance. And again, I think for me, the bottom line here is that there's something about, this is just a hypothesis, it's a speculation at this point, but there's something about the way that synapses work across the spectrum between supernormal, normal, and having some form of cognitive impairment that is being mirrored in the blood. And in order to understand the biochemistry and the metabolism of that is going to take some time, but I think there's a kernel of a new idea there that may reveal, from the perspective of therapeutic targets, a new target or several that we should really learn more about, because it may hold some promise for the ultimate treatment of those individuals who have dementia, cognitive impairment, or are at risk for either of those.
And when you say synapses, you are referring to the connection between neurons, not within the neuron itself, but that relationship that one neuron needs to communicate with the other.
Correct.
Okay.
Right.
Well, and also in your answer, you mentioned this idea of successful cognitive aging.
Yes.
And I know in some of your papers you've actually said there's really a lack of consensus on how to define it.
Yes.
So I'm going to put you on the spot and ask you to define it personally, but then also to tell us how are you living so that your cognition is aging successfully.
Yeah, so I think we're at the stage where we're going to define this as a clinical entity using what might be fairly standardized and validated neurocognitive assessment tools as we do research.
Okay.
In the conduct of life, where we talk about successful aging, we're talking about people who are very robust. They are very engaged socially. They tend to have a full command of many attributes that are part of what we take in every day, whether it be media or conversation. They tend to be very connected. So I suspect, although we haven't really looked at the role of social networks and how these people function in that context, but my surmise would be that they are very well networked. And my sense is eventually, because the one thing that we've done is we did whole genome sequencing, which means we've sequenced the 3.3 billion base pairs of everyone who was followed as part of our study. And eventually, we're interested in being able to map their cognitive function to something that relates to what they inherited from mom and dad, or how it may be modified. We have this whole phenomenon in informational processing that we call epigenetics, where the genome itself doesn't change, but it is modified in a way that, if you will, affects the way it could be read out. So the modification of the genome, we know, is a big part, and this will lead me to what I do, is a big part of what we think lifestyle is modulating. And lifestyle, I would include under that umbrella what we eat, how we sleep, how we exercise, and how we otherwise attend to stress. And in the area of diet, I think that from a personal perspective, you can't see me in the audience, but I'm someone who has a relatively low body mass index, and I have worked to sustain that by daily exercise of a moderate intensity nearly every day. I eat a diet that's Mediterranean-like, trying to make certain that those omega-3 sources are part of the diet, as well as lots of vegetables and fruit. I would also say that although the data are not definitive, but I do think that occasionally a glass of red wine may not be a bad idea. It has to be done in moderation. And then I think finally, sleep is terribly important. There are really interesting new theories about how toxic proteins in the brain are cleared, and it would appear that during certain cycles of sleep, it's very important to have those be commensurate with the need to clear them. And so I find that everyone has a slightly different sleep requirement. I try, whenever possible, to sleep to the max of my sleep requirement. And then finally, stress modulation. I've had some complex jobs where stress goes along with the territory, but I think it's most important to find ways in which you can sort of dissociate from what is immediately in front of you. And I've taken up doing some form of meditation because I think it allows you to really reduce those things that can impinge almost constantly and can engender stress. And I think stress management is something that, unfortunately, in academic medicine, we don't do well enough. And in fact, one of my concerns as a medical educator is that we have a culture that we've promoted now that is associated with more burnout, associated with incomplete management of stress in the lifestyle of someone who might be clinically deployed.
I think it's interesting that you mentioned social networks and how important those
Yes.
are. And in my own clinic, I talk about the importance of social activity.
Great.
But I do wonder, in your research going forward, if you're going to see a difference between generations. Because I look at millennials,
Yes.
and their social networking is much different
Yes.
than our baby boomers.
Yes.
And they're using technology, and they may not be in front of a person like you and I
Yes.
are right now.
Right.
And I wonder how that's going to impact our cognition.
And I don't think we know the answer to that, but my sense is that we should be mindful of evolution. And we've evolved as social species. And the question really is, can you supplant human contact with technology? I have an opinion, it may be wrong, but I think the answer is no. I think human contact is the cornerstone of this sort of social nature of what we do as people. It may well be that in time, the extension of whether it be new forms of virtual reality that sort of mimic the social interactions that we might have, such as you and I talking today on your show, I can't say.
And I would completely agree with you, and I can tell you from talking to other millennials, they would agree that you can't replace a human with a computer or a machine.
I'm on board.
Well then, really I want to end by thanking you for joining us on Dementia Matters, and we really look forward to having you again.
Nate, it's been a pleasure. I really enjoyed talking with you. Thank you.
Thank you. Dementia Matters is brought to you by the Wisconsin Alzheimer's Disease Research Center. The Wisconsin Alzheimer's Disease Research Center combines academic, clinical, and research expertise from the University of Wisconsin School of Medicine and Public Health and the Geriatric Research, Education, and Clinical Center of the William S. Middleton Memorial Veterans Hospital in Madison, Wisconsin. It receives funding from private, university, state, and national sources, including a grant from the National Institutes of Health for Alzheimer's Disease Centers. This episode was produced by Rebecca Wazaleski and edited by Zoe Rugel. Our musical jingle is Cases to Rest by Blue Dot Sessions. Check out our website at adrc.wisc.edu. You can also follow us on Twitter and Facebook at Wisconsin ADRC. If you have any questions or comments, email us at dementiamatters@medicine.wisc.edu. Thanks for listening.